So now we are going to go over the Lineweaver-Burk plot for competitive inhibition. What is a Lineweaver-Burk plot? Well, if we take a look at our Michaelis-Menten plot where we had our substrate concentrations and initial rates, here we have a normal enzymatic reaction and then we have a plot with competitive inhibition, and then we could also have non-competitive and uncompetitive inhibition. The problem with a Michaelis-Menten plot is that we cannot easily distinguish whether an inhibitor is competitive, non-competitive, or uncompetitive because the plots look relatively similar to one another. That is why we use a Lineweaver-Burk plot, because it can help us easily determine whether we have a competitive, uncompetitive, or non-competitive inhibitor. The second reason why we use a Lineweaver-Burk plot is that because in a Michaelis-Menten plot, we have this asymptote which designates Vmax. Now, an asymptote represents a value which we get closer and closer to, but we don't eventually reach. But we want to determine what Vmax is. So what we can do is we can use the Lineweaver-Burk plot to determine our Vmax. So our Michaelis-Menten plot essentially is not useful to see the linearization of data, what our Vmax is, and to determine what type of inhibitor we have. And that is why we have the Lineweaver-Burk plot, which is simply a reciprocal of our initial rates and the reciprocal of our substrates. So on our x-axis, we have 1 over substrate concentrations, and our y-axis, we have 1 over our rates. Now, we can make note that for a competitive inhibitor, this is our plot, whereas for a normal enzyme, this is our plot. Two things we can make note of. First of all, we need to understand our equation for a Lineweaver-Burk plot. A Lineweaver-Burk plot is a linearization of our data that we had in our Michaelis-Menten graph. A linearization of data includes this equation, which we are almost all familiar with, and that is y equals mx plus b, where y is our y values, x is our x values, b is our y-intercept, and m is our slope. But we can write this equation in the format for the Lineweaver-Burk plot, where 1 over initial rates is going to be our y values, 1 over initial rates. We have our slope, which is km over Vmax. And then we have our x values, which are going to be 1 over our substrate concentrations. And then we have our y-intercept, which is going to be our 1 over Vmax. Now, it's really important to understand over here that, remember, in competitive inhibition, our Vmax remains the same, and our Vmax is our y-intercept. So right over here is our y-intercept, and we can actually see in our Lineweaver-Burk plot that for a normal enzymatic reaction and when a competitive inhibitor is present, our Vmax remains the same. Now, we can make note that our Km has changed because our Km is our x-intercept. Remember that in order to find our x-intercept, we simply denote our y value being equal to 0, and then we solve for x. In this case, we solve for our 1 over substrate concentration, in which we find that our Km is going to be equal to our x-intercept value, which is right over here. And we can make note that Km has increased when we have competitive inhibitor present. We can also note that our slope has increased. Why is our slope increased? Because slope is Km over Vmax. If we are increasing Km and our Vmax stays the same, our overall slope will increase. So in competitive inhibition, we will see a steeper slope compared to when there is no inhibitor present. And remember, our Km is changing. What is Km? Km is our substrate concentration at 1/2 Vmax, and our Km is increasing because we require more substrate to reach our 1/2 Vmax. And that is essentially the Lineweaver-Burk plot for competitive inhibition. It takes the reciprocal of our initial rates and our substrate concentrations, and it linearizes the data so we can easily distinguish whether we have competitive inhibition or we have other forms of inhibition. And we can use the Lineweaver-Burk plot to determine our Vmax. If we wanted to determine the Vmax, we would simply take a look at what our y-intercept was on our Lineweaver-Burk plot equation, and then we would take the reciprocal, because remember, this is 1 over Vmax, and if we take the reciprocal of this value, we can determine Vmax. Same thing for our Km values. Km is our x-intercept, so we can take a look at our x-intercept values over here, take the reciprocal of them to figure out our substrate concentrations, and that will be our Km. And for competitive inhibition, Km is increasing because we require more substrate to reach our 1/2 Vmax.